Those were examples of the non-specific, the innate immune response. Non-specific means that it's not targeted at one particular type of bacteria or virus or whatever it may be. And innate means that that is something that you have, it's there to begin with when you're born, actually before birth, and it's always there and ready as long as there's nothing wrong with your immune system. The specific responses, on the other hand, are much more targeted precisely. This identifies not, this is some sort of gram-positive bacteria. This says that's measles virus. Something very specific that has been identified as being a problem to the body. It's also referred to as acquired because this is not something that you're born with. Instead, your immune system has to encounter this particular thing and realize it's a problem and develop a response to it. So it's somewhat slower the first time, but then once you know, you are ready to respond quickly the next time. So the specific immune response characteristically will develop over several days at first in response to a very specific pathogen. Once you've got it, then it is activated again very quickly. So the chances of getting sick from it again are rather small. And this relates to a special set of genes known as the major histocompatibility complex, MHC, used to tell, is this me or something else? Definitely would not have been a good thing for Mendel to try to start with, figure out how it works, because there's over 40 alleles for each of multiple loci. Of course, also would have required chemistry that Mendel didn't have available at the time. And so, with so many different alleles, multiple loci, the chances of any two people having the same exact set of these proteins is very, very small. Basically, only identical twins would. So, this means that your cells have a unique combination of the major histocompatibility complex proteins on their surfaces. And so your immune system has encountered those and knows, okay, these are the things that I should be seeing in the cells that are me. If I see something else, this is a cell that is not me, this is a problem. Now, of course, these days, medically, we can have organ transplants, blood donations, things like that, but that's bringing something from somebody else. We have to pay attention to various histocompatibility things like blood type in order to make sure that the things are compatible. The way one important part of the acquired immune system develops, we have certain cells that are phagocytic. They will swallow and break down things they encounter. And so this will include, say, bacteria. Bacteria have their own proteins on their surface, things like that. And so the cell encounters it, breaks it down, and then puts out on its surface these random molecules that it found in what it digested. And then these chemical bits, if another immune cell encounters this and, wait, I recognize this, that's something bad, that will trigger a response by the immune system. Similarly, the immune system producing antibodies, if these bind to some sort of chemical, typically it will be a protein on the surface of some particle, then the antibodies attach those, and that triggers a further immune response. Looking at more detail in this process, the B cells, these are one special type of our white blood cells, leukocytes, and these are the ones that make antibodies. And the genes to make antibodies have very high mutation rates, so you're always coming up with new types of antibodies in this process. Okay, and a bunch of them don't do anything. And so, okay, well, that one wasn't useful. But if one of those antibodies happens to match and stick onto something that shows up in the body, and it's identified as a pathogen, then the B cell, instead of just, oh, here's another antibody, see what happens. Yep, I found something. Alert, this is something important. So the B cell divides lots of times, so there are lots and lots of B cells making that same exact antibody. And so these antibodies, they can be floating loose in the blood or lymph system. Also, they can be attached onto immune cells. Some of these new activated B cells will go into storage as the memory B cells are ready for next time, while the others are cranking out all those antibodies. The antibodies, again, are molecules that can bind to these other things. And for one thing, bind to that other thing, that's covering it with antibodies. That's not going to be good for whatever that was going to do, whether it's a bacteria that's now got all this stuff stuck on it, or maybe it's a toxic chemical, but now it's got other stuff stuck on it, and it can't do its poisoning. Also, if you've got something with an antibody stuck on it, there are immune cells that come along and say, hey, this has got an antibody, it must be bad, destroy it. And so those other cells will attack things that have antibodies stuck on them. Antibodies can also be used quite extensively in research. They can detect very tiny amounts of a specific protein. So for example, they're used in pregnancy tests, detecting proteins that will be present in response to early pregnancy. Once the immune system has identified a problem, not just B cells, but various other types, the memory T cells also will be stored in various parts of the body, not just the lymph system where they tend to start out, but parts where there's often infection. Lung, intestine, those are places that are encountering stuff from outside. The liver and kidney have key functions in filtering, cleaning blood, picking up stuff out of the blood system, and so they're also likely to pick up things. Good place to have good defenses. If whatever that is shows up again, the memory T cells will activate, become the TC cells, churning out chemicals to kill invading bacteria and the like. Likewise, our memory B cells will turn on a special gene saying, stick around. Don't go through cell death, apoptosis. And so they're still producing low levels of antibody after the initial infection has been dealt with. And if it shows up again, they're ready to go. So that first encounter with whatever stuff it is, we have the gradual buildup of the primary immune response of the specific immune system. And so these antibodies takes on the order of 3 days to 2 weeks for the antibody production to get going. Once it's started, it goes rapidly. Those B cells are dividing and making more quickly. After that's dealt with that infection, the memory cells can last in your body for many, many years. Eventually, a boost can help. There are a number of vaccines, for example, where you're supposed to get a booster after a while to make sure the cells are still maintaining their thing. Once you have those cells all ready to go, however, if you encounter that same antigen once more, the body is able to crank up the production quite quickly. It doesn't have to be finding the antibodies already got, it just has to make lots more of those B cells, and the response is much quicker, and you have very little problem with it the next time. So you develop active immunity. You've been exposed to the antigen, your body has identified it, found the antibodies, and is storing the memory B cells ready to go. Now, of course, this happens if you do get sick or otherwise encounter one of these things, but it can also be given by a vaccine, by immunization. And so this triggers your immune system. You have, say, a weakened version of the disease-causing organism, or perhaps dead ones, or perhaps something that's similar but doesn't make you sick the way the real problem one does, or perhaps a somewhat altered version of molecules produced by the pathogens. And so you have a non-dangerous exposure to these hazards, and your immune system comes around, what's this stuff? Hey, I've got an antibody that matches, attack this. And so the B cells get going, you get your memory B cells that have the antibody already. And now, thanks to having gotten the shot, you did not get particularly sick from getting a shot. Sometimes it hurts for a little bit, or mild symptoms, but that's it. Now, when the real bad guy comes along, you're already prepared. You've got your B cells that already know how to make the antibody, and so as soon as you encounter it, then your body quickly ramps up the production and can stop that infection in its tracks. As opposed to if you hadn't had the vaccine, your body doesn't recognize this yet, and so, we seem to be starting to have a problem here. Any B cells got something matching this yet? Anybody? While your body is doing that, the virus or bacteria, whatever problem is multiplying. But if you've already encountered it through the vaccine, your body responds much quicker and stomps out that vaccine faster.